Hello student, welcome to EPG Pathsala. I am Dr. Indrakant K. Singh from Department of Zoology, Deshbandhu College, University of Delhi. Today we are going to discuss about cell cycle part 1, that is on mitosis. In this module we are going to discuss about the historical background of cell cycle, basic introduction of cell cycle, and very importantly about the DNA and chromosomes. As the process of cell division differs among the prokaryotes and eukaryotes, so we will discuss it separately. Eukaryotic cell division is a complex and highly regulated event. We will focus it in detail. Now, let us see what we are going to learn from this module. I will explain you first about the historical aspect of the cell cycle. Then, we will try to understand the phases of cell cycle. Further, we will come to know about the types of cell cycles. Also, we are going to discuss the different stages of mitosis in this module. We will cover the significance of mitosis, and at the end of this module, I will summarize whole cell cycle, especially part 1, that is mitosis. You all must be wondering, where do cells come from? How a single cell zygote formed by a fusion of sperm and ova can give rise to a complete multicellular organism? How does a cell divide? So, before going in much detail of cell cycle, you should be clear about what is cell cycle. Cell cycle is a series of events that finally led to division of parent cell, giving rise to two daughter cells identical to parent cell. These events are highly regulated and well organized to maintain cell integrity. Growth and reproduction are characteristics of cells. All cells reproduce by dividing into two, with each parental cells giving rise to two daughter cells each time they divide. Cell cycle mainly involves two main processes. Number 1, cell growth, and number 2, cell division. Cell growth is increase in cytoplasmic content, and cell division is the process when cell finally divides into two newly formed daughter cells. Now, you must be thinking, why do cell divide? What is the purpose of cell cycle? Is it same in both prokaryotes and eukaryotic cell? No. So basically, cells divide to produce new cells for growth as well as replacement of damaged old cells. Cell division differs among prokaryotes including bacteria and eukaryotes including protista, fungi, plantae and animalia. There are two major phases of cell cycle. Number 1, DNA replication, and number 2, cell division phase. In DNA replication phase, there are involvement of copying of the parental DNA, and in cell division phase, in which cell divides the cell component equally into two daughter cells. So, for the normal development and maintenance of the multicellular organism, coordination of cell division is very crucial across different tissues and organs. It can be achieved by coordinating timing of cell division as well as the regulation of cell division rate. It would be interesting to know that all cells of our body cannot follow the same cell cycle. Any kind of failure in these crucial events ultimately led to cancerous growth. Since the discovery of third tenets of cell cycle proposed by Carl Virchow, new cells arise from pre-existing cells. Cell cycle becomes the subject of intensive research. For the first time, Oscar Hartwig described meiosis in sea urchin in 1876. 4 years later, Walter Fleming gave the term mitosis. The advancement of molecular and genetic techniques in 20th century strengthened our understanding of cell cycle and its regulatory mechanism, which we are going to discuss in next few minutes. In 1974, mechanism of cell cycle switching between proliferative and G0 phase of cell cycle was discovered. Later on in 1980, various new discoveries came into light like discovery of centromeres from budding yeast, all checkpoints and their controlling mechanism, etc. In 1990, scientists identified cyclin-dependent kinase, that is a new class of protein, and CDK inhibitors. Recent discovery, which was reported in beginning of 21st century, three scientists, Leland Hartwell, Tim Hunt, and Paul Nurse, were jointly awarded Nobel Prize in Physiology or Medicine for their discoveries of key regulators of the cell cycle. Now, you must be thinking, do all the cells show same cell division? Do all the cell divides throughout their life? Is there any connection between cell division and generation of variants? All your queries will be clear very shortly. Frequency of cell division varies among the different cell types. For example, cells of embryo divides approximately in 20 minutes. Our skin cells shows frequent cell division around 12 to 24 hours per cycle. They are replaced very fastly. If you take example of our liver cells, which retains its ability to divide only when they require. Few of our cells, like mature muscle cells and nerve cells, permanently enter in the G0 phase and stops dividing after maturity. Before going in detail of cell cycle, knowledge about DNA, which is known as instructor of cell, and chromosome, which is also known as carrier of genetic information, is very essential. All the information required for making a cell parts or complete organism resides in DNA. So, before cell cycle, it has to be replicated so that newly formed daughter cells get its own complete set of DNA molecules. Chromosomes are the thread-like structures present inside the nucleus. They are designated as the carrier of hereditary information. Its structure differs among prokaryotes and eukaryotes. In case of prokaryotes, they have single circular chromosome attached to the inside of cell membrane, and in case of eukaryotes, they have around 10 to 50 chromosomes. Few plants have even more than 50 chromosomes. Few terms should be clear to you before discussing cell cycle. You can see in the given figure of chromosomes, it is showing chromatids, sister chromatids, and centromere. Chromatids are the duplicated chromosomes formed after DNA copy. When a single chromosome replicates and forms two identical chromatids, they are termed as sister chromatids. These sister chromatids are held at centromere. Very clearly, you can see these structures in given figure also. First of all, we will focus on prokaryotic cell division. Prokaryotes are similar in their organization compared to eukaryotes. Prokaryotic chromosomes is a single DNA molecules. During prokaryotic cell division, firstly, the DNA replicates and forms its copies. Then, each copies of DNA is attached to different parts of cell membrane. Then, gradually, cell begins to pull apart, involving cross-wall formation. And finally, cell splits, leading to cytokinesis. As a result of prokaryotic cell division, two genetically identical cells are formed except rare chances of spontaneous mutation. Now, I will come to eukaryotic cell cycle. It is complex compared to prokaryotic cell cycle. It involves highly regulated and well-organized series of events or phases. Major fundamental events common to all eukaryotic cells are chromosomes replication. This phase involves DNA replication, and chromosomal segregation, which involves equal segregation of parental DNA. Variety of heterodimeric protein kinases are involved in cell cycle. They are key regulators of cell cycle. These heterodimeric protein kinase includes number 1, cyclin, which is regulatory subunit, and CDK, that is cyclin dependent kinase, which has catalytic subunit. These proteins regulates the activity of many proteins by phosphorylation at specific regulatory sites. They can also coordinate the activity of these proteins involved in cell cycle by activating some and inhibiting others. Now come to phases of cell cycle. These are broadly divided into two phases: interphase and M phase. Interphase is the period between two cell division, which is further divided into G1, S and G2. In G1 phase, which is known as gap 1, this gap is immediately after cell division phase. S phase, which is known as synthetic phase, which involves DNA synthesis. G2 phase is known as gap 2. This gap is between S phase and M phase. M phase, that is the mitotic phase, it involves final phase of cell division, which is further divided into four phases: prophase, metaphase, anaphase and telophase. So, interphase involves cell growth and various metabolic activity. M phase is the actual division phase. You can clearly see all these phases in given figure. As we have seen earlier, interphase is further divided into G1 phase, S phase and G2 phase. Let's start with G1 phase first. It is the interval between mitosis and beginning of DNA replication. During this phase, cells is metabolically very active but does not replicate its DNA. Cell basically prepare itself for DNA replication by synthesizing RNAs and proteins. After a preparatory gap phase 1, cell enters into another phase that is known as S phase. During this phase, parent cells replicates its DNA. Important point which has to be kept in mind is that only amount of DNA double per cell, but number of chromosomes remains the same. Now, cell enters into another gap phase, that is G2, in which cell do all preparation needed for cell division. For example, centrioles is made, synthesis of proteins and cell organelles. This phase is immediately after DNA replication phase. Let's quickly summarize all the events occurring during interphase of cell cycle. It involves replication of parental DNA, replication of centrioles if present. As few animal cells like early mouse embryo, cells of higher plants lack centrosome, still all these can form bipolar mitotic spindle. Ultimately, cell prepares its machinery for M phase, that is actual division phase. In eukaryotic cells, majorly two types of cell division is observed: mitosis and meiosis. In case of mitosis, two identical daughter cells are produced as a result of cell division. These daughter cells have identical genetic makeup. It is rarely produce any variations. But in case of meiosis, 4 daughter cells are produced, which are dissimilar to parents. These daughter cells have half genetic material of parent cell. It leads to generation of variants. So let's begin with mitosis. This type of cell division is observed in both somatic as well as germ cells. It involves faithful segregation of replicated DNA molecules of each chromosomes into 2 nuclei, and daughter cells, which are formed, are identical to their mother cells from which they arose. It was discovered by Strasburger in plant cell and by Walter Fleming in animal cells. The process of mitosis is crucial to all eukaryotic organisms. In some single-celled organisms, such as protozoans and some fungi and algae, mitosis provides the basis for sexual reproduction. Multicellular diploid organisms begins life as single-cell fertilized eggs called zygotes. The mitotic activity of the zygote and subsequent daughter cells is the foundation for the development and growth of the organisms. In adult organisms, mitotic activity is the basis for wound healing and other forms of cell replacement in certain tissues. For example, the epidermal cells of skin and intestinal lining of humans are continuously sawed off and replaced. Mitosis involves 2 events: karyokinesis and cytokinesis. In case of mitosis, the genetic material is partitioned into daughter cells. During nuclear division, or you can say karyokinesis, this process is quite complex and require great precision. These chromosomes must first be exactly replicated and then accurately partitioned. Karyokinesis is followed by cytoplasmic division or cytokinesis. This less complex process requires a mechanism that partitions the volume into 2 parts, then encloses each new cells in a distinct plasma membrane. Mitosis is generally divided into 5 stages. Number 1, prophase, prometaphase, metaphase, anaphase, and telophase. We will discuss these stages separately in detail. First, we will start with prophase. During this phase of mitosis, DNA, which was earlier present in the form of chromatin, start condensing to form chromosomes. Over half of the mitosis is spent in prophase, a stage characterized by several significant occurrences. One of the early events in prophase of all animal cells is the migration of 2 pairs of centrioles to opposite ends of the cell. These structures are found just outside the nuclear envelope in the area of differentiated cytoplasm called centrosome. Each pair of centrioles consists of 1 mature unit and a smaller, newly formed centrioles. The centrioles are responsible for organizing cytoplasmic microtubules into the spindle fibers that run between these poles, creating an axis along which chromosomal separation occurs. As the centrioles migrate, the nuclear envelope begins to break down and gradually disappears. In a similar fashion, the nucleus disintegrates within the nucleus. While these events are taking place, the diffuse chromatin fibers have begun to condense until distinct thread-like structures, the chromosomes, becomes visible. It becomes apparent near the end of prophase that each chromosome is actually a double structure split longitudinally except at a single point of constriction, the centromere. The 2 parts of each chromosomes are called sister chromatids because the DNA content in each of them is genetically identical. Sister chromatids are held together by a multi-subunit protein complex called cohesin. Condensin is a multi-protein complex associated with the chromosomes, causing its compaction. It forms a ring around supercoiled loops of DNA within chromatin. Cohesin plays a role in holding DNA of sister chromatids together. So, as you can see in figure also, condensin is an intramolecular cross-linker and cohesin is an intermolecular cross-linker, ultimately holding chromatids of each mitotic chromosomes firmly at their centromeres and relatively loosely along their extended arms. Now, we can focus on how mitotic spindles are formed. During G1, the centrosome contains a single pair of centrioles that is no longer as tightly as they were during mitosis. During S phase, daughter procentrioles form adjacent to maternal centriole so that two pairs of centrioles becomes visible within the centrosome. At the beginning of mitosis, centrosome splits. As they separate, the centrosome organize the microtubules fiber that make mitotic spindle. You'll be amazed to know that formation, even a single additional centriole, leads to development of cancer. During early prophase, microtubules appears in a sunburst arrangement, or you can say ester, around each centrosome. Microtubules grow by addition of tubulin subunit to their plus end, while their minus ends remain associated with the centrosome. Later on, centrosome separates from one another towards opposite ends of the cells. As we have discussed earlier, animal cells and cells of higher plants lack centrosomes. In these cells, mitotic spindles are nucleated near the chromosomes rather than at the poles where centrosome resides. Cyclin B, that is CDK1, plays important role in breakdown of nuclear envelope at end of prophase. Now, let's quickly glance all the events that takes place during prophase. So, you can see in prophase diagram also, DNA in the form of chromatin condenses to form chromosomes and it becomes visible under light microscope. Mitotic spindles are formed, which is required later on. Nuclear membrane and nucleolus disappears by the end of prophase. Finally, chromosomes are prepared for segregation. The term prometaphase refers to the period of chromosome movement. You will be amazed to know that few of the cell organelle like mitochondria, lysosome, peroxisome and chloroplast of the plant cell remain intact through mitosis. Dissolution of nuclear envelope marks the beginning of next phase, that is prometaphase. In this phase, chromosomes are directed towards the equatorial region of cell and microtubules that makes contact with kinetochores are captured. You can see in the figure where it is depicting how DNA is compacted in the form of chromosomes during the prophase. This compacted form of DNA is easy to segregate between daughter cells. You can see how exactly chromosomes look like. Mitotic chromosomes are composed of similar types of fibers as seen by electron microscopic examination of whole chromosomes isolated from mitotic cells. It appears that chromosomes compaction does not alter the nature of chromatin fiber, but rather the way that the chromatin fiber is packaged. Examination of sections through a mitotic chromosome reveals the presence of a proteinaceous button-like structure that is called kinetochore, At the outer surface of centromeres of each chromatid, the kinetochore assembles at the centromere during prophase, and it serves as the site of attachment of chromosome to the dynamic microtubules of the mitotic spindle. This also serves as the residence of several motor proteins involved in chromosomes mobility and a key component in the signaling pathway of an important mitotic checkpoints. Two proteins, NDC80 and CNEPE, plays important role in mitosis during prometaphase. NDC80 kinetochore complex is an essential kinetochore protein which binds the surface of adjacent microtubules by forming fibrils. If the chromosomes are not properly attached from both the poles, it can trigger prometaphase arrest. So, basically it is involved in spindle checkpoint signaling. CNEPE, which is known as centromere associated protein E precursor, belongs to kinesin superfamily. It is required for chromosomal stability. It is a key player in the chromosome's movement towards metaphase plate. It provides flexibility and motile linking of kinetochore. It prevents aneuploidy by serving as mitotic checkpoint signal. CNEPE also serves to power chromosome congregation. As we have discussed that microtubules are continuously growing and sinking at their plus end, then how kinetochores are able to maintain their attachment to the microtubules? In reality, a kinetochore makes initial contact with side wall of microtubules rather than its end. Kinetochores have a tendency to become stably associated with the plus end of a spindle microtubule. The chromosomes of a prometaphase cells are moved by process called congregation towards the center of the mitotic spindle, midway between the pole. These forces required for chromosomes movement during prometaphase are generated by motor proteins associated with both the kinetochores and the arms of the chromosomes. As chromosomes congress towards the center of the mitotic spindle, the longer microtubules attached to one kinetochores are shortened, while the shorter microtubules attached to the sister kinetochores are elongated. These changes in microtubules length are thought to be governed by differences in pulling force, or you can say tension, on the two sister kinetochores. Shortening and elongation of microtubules occurs primarily by loss or gain of subunit at the plus end of the microtubule. Once all of the chromosomes have become aligned at spindle equator, with one chromatid of each chromosome connected by its kinetochore to microtubules from one pole and its sister chromatids connected by its kinetochore to microtubules from the opposite pole, the cell has reached the stage of metaphase. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. The mitotic spindle of the metaphase cell contains a highly organized array of microtubules that is ideally suited for the task of separating the duplicated chromatids positioned at the center of the cell. Functionally and spatially, the microtubules of the metaphase spindle of an animal can be divided into three groups. Number one, astral microtubules, which radiates outward from the centrosome into region outside the body of the spindle. They help to position the spindle apparatus in a cell and may help to determine the plane of cytokinesis. Second, that is chromosomal microtubules, which extends between the centrosome and its kinetochores of the chromosomes. In mammalian cells, each kinetochore is attached to a bundle of 20 to 30 microtubules, which forms a spindle fiber. During metaphase, the chromosomal microtubules exert a pulling force on the kinetochores. As a result, the chromosomes are maintained in the equatorial plane by a tug-of-war between balanced pulling forces exerted by chromosomal spindle fibers from opposite poles. During anaphase, chromosomal microtubules are required for the movement of chromosomes towards the pole. Third type of microtubules, that is polar microtubules, also called interpolar microtubules, which maintains the mechanical integrity of the spindle. Even though the microtubules appear stationary at this stage, injection of fluorescently labeled tubulin subunits indicates that the component of the spindles are in a dynamic state of flux. Subunits are incorporated preferentially at the kinetochore of chromosomal microtubules and the equatorial ends of the polar microtubules, and are lost preferentially from the minus end of the microtubules in the region of the poles. Tubulin subunits move through the microtubules of the metaphase spindles at the rate of about 1 micrometer per minute. You must be thinking how subunits move along the chromosomal microtubules from kinetochore towards the poles. As more subunits are added to the plus end than are lost, there is a net addition of subunits at kinetochore. Meanwhile, the minus end of the microtubules experience a net loss, and thus subunits move along the chromosomal microtubules from the kinetochore towards the pole. Let's quickly revise major events during metaphase. During metaphase, chromosomal attachment to the mitotic spindle is established through kinetochore, and chromosomes are aligned along the metaphase plate at the equator of the cell. You can clearly see these changes in the given figure. Now, after alignment of the chromosomes along the equator, these chromosomes are ready to separate towards the opposite poles by the splitting of sister chromatids of each chromosomes. This phase is known as anaphase. Proteolysis also plays a major role in this cell cycle progression. Various biochemicals and genetic approaches have been applied to gather information on the mechanism responsible for the initiation of anaphase. Anaphase is controlled by two distinct multiprotein complexes, SCF and APC. They add ubiquitin to proteins at different stages of cell cycle, targeting them for destruction by proteasome. APC has two versions differing in the types of adapter, containing either a CDC20 or CDH1 adapter protein. These adapter proteins alter substrate recognition by APC complex. SCF, known as SKP choline F box containing complex, is majorly active during interphase, and APC, which is known as anaphase promoting complex, is primarily active during mitosis. And G1 phase pointed out earlier that two distinct multiprotein complexes, SCF and APC, add ubiquitin to protein at different stages of cell cycle, targeting them for destruction by a proteasome S. SCF acts primarily during interphase. In contrast, anaphase promoting complex or APC plays a key role in regulating events that occur during mitosis. The APC contains about a dozen core subunits, in addition to an adapter proteins that play a key role in determining which proteins serve as a APC substrate. Two alternative versions of these adapter proteins, that is CDC20 and CDH1, play an important role in substrate selection during mitosis. APC complexes containing one or other of these adapters are known as APC CDC20 or APC CDH1. APC CDC20 becomes activated prior to metaphase and ubiquitinates a major anaphase inhibitors called sequirin. So, named because it secures the attachment between sister chromatids. The ubiquitination and destruction of sequirin at the end of metaphase release an active protease called separase. Separase then cleaves a key subunit of the cohesin molecules that hold sister chromatids together. Cleavage of this cohesin triggers the separation of sister chromatids to mark the onset of anaphase, as shown in the figure. SAC, that is spindle assembly checkpoints, inhibits APC CDC20 from eliciting anaphase until all replicated chromosomes become properly aligned at metaphase plate region. Near the end of mitosis, CDC20 is inactivated and the alternate adapter CDH1 takes control of APC substrate selection. This enzyme completes the ubiquitination of cyclin B. Destruction of cyclins led to precipitous drop in activity of mitotic CDK, that is cyclin B CDK1, and progression of cell out of mitosis and into G1 phase of the next cell cycle. Now, question is how a cell determines whether or not all chromosomes are properly aligned at a metaphase plate. Basically, the presence of MAD2, which is a main controller of spindle assembly checkpoint at those kinetochores which are not attached to microtubules, sends a wait signal to cell cycle machinery that arrests cell cycle into anaphase. Colchicine, which is commonly used in cancer treatment, is a mitotic inhibitor drug. It can disrupt microtubules formation, which plays important role in chromosomal segregation. Now, let us quickly revise major events during anaphase. Basically, during this phase, centromere divides, causing pulling apart of sister chromatids towards the opposite poles through spindle fibers. So, finally, cells are provided with identical set of genes, as you can see in the figure. As the chromosomes reach their respective poles, they tend to collect in a mass, which marks the beginning of the final stage of mitosis or telophase. During telophase, daughter cells return to the interphase conditions. Means the mitotic spindles disassembles, the nuclear envelope reforms, and the chromosomes becomes more and more dispersed until they disappear from view under the microscope. The actual partitioning of the cytoplasm into two daughter cells occurs by a process to be discussed shortly. Karyokinesis, nuclear division, is followed by cytokinesis, which is division of cytoplasm. Cytokinesis is the final stage of mitosis. It differs between a plant cell and an animal cell. In case of plant cell, it starts in the center and then move towards periphery, meaning towards lateral wall. But in case of animal cell, a cell furrow appears during the anaphase in the plasma membrane, which gradually deepens. Finally, at the end, it led to formation of two daughter cells having own genetic material. In case of animal cells, plane of cytokinesis is the same as occupied by chromosomes during the metaphase. One theory which was proposed by Douglas Marsland in 1950, known as contractile ring theory. It states that force necessary for cytokinesis is produced in the thin band of cortex. Cortex is a portion of cytoplasm present beneath the plasmamembrane of the fur. Microscopic studies of cortex revealed the presence of bulky of actin filament along with small number of short and bipolar myosin filament. Location of the cleavage furrow is determined by anaphase mitotic spindle by activation of rho A in a thin ring inside the cortex region. As we have already discussed that in a plant cell wall formation starts in the center and grows laterally, finally divides the cell. Unlike animal cell division, plane of cell plates formation is perpendicular to the plane occupied by mitotic spindle. What would happen if karyokinesis is not followed by cytokinesis? You can see in figure that if process of cytokinesis is disturbed, then instead of two daughter cells, only one daughter cell will form, having number of chromosome doubled. Now, we will discuss about the significance of mitosis. First, as mitosis takes place in all somatic cells, the daughter cells which are formed have constant chromosome number and they are identical to parent cell. It has very negligible chances of variation. Mitosis mainly takes place in those cells where growth and development is required, like in zygote. It is even helpful in wounds repair, and it is an important mode of asexual reproduction. Finally, let us now summarize what we have learned in this module so far. Cells reproduction involve copying and separation of their genetic information and then dividing into two daughter cells. The cell cycle phases are interphase and M-phase. Interphase, where the cell is majorly involved in cell growth and in diverse metabolic activities, that is further divided into G1 phase, where it involves cell growth and cell prepare itself for copying its genetic material. In S phase, DNA synthesis takes place. In G2 phase, involve various biochemical events necessary for cell division. Second phase, that is M phase, which includes mitosis. It is further divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. During the mitosis, there is a very important event that is called cytokinesis. As a result of mitosis, two genetically identical daughter cells are produced having same number of chromosomes. Cell cycle is highly regulated process controlled at various checkpoints by interaction between cyclin and cyclin dependent kinases. Now, I would briefly explain basic concepts. The structure of cell is elaborate and complex, with most components involved directly or indirectly with genetic processes. In diploid organisms, chromosomes exist in homologous pair, where each member is identical in size, centromere replacement, and gene loci. One member of each pair is derived from the maternal parents and other from the paternal parents. Mitosis and meiosis are mechanisms by which cells distribute the genetic information contained in their chromosomes to progeny cells in a precise and orderly fashion. Mitosis, which is one part of the cell cycle, is subdivided into discrete stages that initially depict the condensation of chromatin into diploid number of chromosomes. Each chromosome first appears as double structure, consisting of pair of identical sister chromatids joined at a common centromeres. As mitosis proceeds, centromeres splits and sister chromatids are pulled apart by spindle fibers and directed towards the opposite pole of the cell. Cytoplasmic division then occurs, creating two new cells with identical genetic information containing in the progenitor cell. Thank you.